Good day to everyone. I am Dr. M. Gopi, Assistant Professor and Head, Department of Plant Biology and Plant Biotechnology, Guru Nanak College, Chennai. Today, I will discuss about plant microtechnique. What is plant microtechnique? Microtechnique is the ancient plant biology. You have to take the cross sections of the plants, that is called it as plant microtechnique. Introduction. Plant anatomy plays an important role in understanding of the plant biology, and it is a realistic interpretations of morphological, physiological and phylogenical aspects of all the plant body. It must be based on a thorough knowledge of the structure of most of the cells and tissues. Definitions. So, the microtechnique, it is a tissue preparation for almost all microscopic examinations. There are different methods are used so far. However, the basic principles are same. It usually involves hardening of the tissues and followed by sectioning, that is cutting. It involves two phases. First phase is paraffin techniques, second phase is freeze techniques. Paraffin technique, it will take 14 days, whereas freeze techniques, it will take immediately. This is the difference between these two. Historical background. When you talk about the microtechnique, microscope, it is one of the aid to identify the plant organs. The microscope, it is first identified or invented by the Zachariah Janssen during 1590s. It is based on the Galileo telescope, that is called it as light microscope. Light is the source of illumination, that is why it is called it as light microscope, or otherwise call it as simple microscope. And then it is followed by compound microscope developed by Anton van Leeuwenhoek. He has identified the simple creatures like protozoa, bacteria, red blood corpuscles. During 1665, Robert Hooke identified some of the slices of the cork cells, call it as cells. And then 1855, Rudolf Virchow identified the cells it is developed from pre-existing cells and it consists of chemical compositions. It is react with each other, that is why it is called it as metabolic activities. And then Matthias Schleiden and Theodor Schwann during 1883 explained about the cell theory. All the living organisms is having the cells. The cell is the basic unit of all living organisms. And then the microtechnique, it is derived from two Greek words. One is mikros, another one is kobos. So, mikros means it is a small, the kobos means lookout. That means we are looking the small objects, that is called it as microtechnique. If you are handling the plants, so it is called it as plant microtechnique. The microtechnique, it consists of two steps or two phases. First step, preparation of the thin and transparent sections. The next step, it is immersed in stains. These two steps involved for plant microtechniques. What are the various steps involved in plant microtechnique? The first one, the exploration and collection of plant material from different forest areas, and it is ideal with the preservation. The preservation is called it as fixation of the plant material. So, the chemicals used for fixing the plant materials, it is ideal usually FAA, formalin acetic acid alcohol. And then the freehand sectioning of the plant material or microtome sectioning of the plant material. The first one freehand section, second one we are using the instrument, that is called it as microtome. And then followed by infiltration of plant materials with using the paraffin wax and dewaxing. Microtome section is having two types. One is sledge microtome, another one is rotary microtome. Rotary microtome, it is useful for herbaceous material, whereas the sledge microtome, it is used for hardened materials, for example wood. And then followed by staining procedures. It may be single staining or double staining for the plant materials. And then it may be temporary preparation or permanent preparation or semi-permanent preparation, depends upon the requirements. The third category, maceration of plant materials. So, the maceration, it is a technique you can isolate the elements from all the living organisms. The last one, micrometry. You have to identify and measure the material size with everything, that is called it as micrometry. Next one, scope of the plant micro technique. So, what is the use of the plant micro technique? The knowledge of plant anatomy is also essential to solve many important everyday problems. The next one, identification of the unknown things, and then food contaminations, adulterants, the finally even forensic problems is also solved every time. That is why it is the scope of plant micro technique. Next one, aim of the plant micro technique. The main aim of the plant micro technique of this program is to introduce students to some useful techniques in the study of plant structure. At the same time, they will also learn the basic anatomical organisms of plant organs as well as cell and tissue characteristics. Aim of fixing and killing, this is the first step of plant micro technique. Killing or mixing of the plant materials are the preliminary important process in plant micro technique. The term killing means the sudden and permanent termination of the life process of individual cells of which the plant body is made up of, to stop the physiological activity of living things, to preserve the cell structure, cell wall, nucleus, chromosome and other cell organelles without any alteration in structure, to increase the refractive index of the cytoplasm for the microscopic studies. Killing and fixing are two distinct processes, but both are usually carried out by means of a single fluid or reagent, commonly a mixture of various chemical reagents. Fixative formulas. How to prepare fixative? Fixative is a chemical substances. One of the best substances is carnise fluid. How to prepare carnise fluid? Acetic acid, alcohol mixture. Why we are choosing this particular fixative? This fixative, it is easily act on the materials because of the great penetration power of each. For root tip, 15 minutes is enough for the fixation, whereas anther we need to preserve for 1 hour. Leaving the material for more than adequate time will lead to the production of artifacts such as cytomixing, mixing of chemical material as well as the materials. And carnise fluid is useful for cytological studies, for example, mitosis and meiosis. Next, carnise fluid. How to prepare carnise fluid? So, we have to prepare in two ways. One is 100% ethyl alcohol for 15 ml, glacial acidic for 5 ml. The next method, 100% of ethyl alcohol for 30 ml, glacial acetic acid 5 ml and then chloroform 15 ml. This is the preparation methods, that is called it as carnise fluid. Fixative formulas. The next example, FAA. What is FAA? Formalin, acetic acid alcohol mixture, and then this fluid is popularly known as FAA and is a standard fixative of botanical micro technique since it is used more extensively than any other reagent. How to prepare this FAA? So, ethyl alcohol 50% or 70%, 90 ml, glacial acetic acid 5 ml, formalin 5 ml. FAA may be used with almost any plant material intended for anatomical or morphological study. Material may be left in its almost indefinitely without any damage. That is why FAA is very ideal for studying. FAA is the ideal fluid to take on longer collection through tip. It is a conclusion part. So, it is the structural organization, it is very much useful to identify the plant parts as well as it is correlated with functions of the all the living things, especially plant organs. So, that is why micro technique, it is a vital part of studying the plants kingdom during the research. So, thank you.